Hello and welcome to this video on the novel and prospectively best alternative to animal research, organ on a chip. This is a relatively new technology which recreates the function, response and mechanisms of individual organs or organ systems. It is almost an artificial organ but for the size and a certain degree of complexity. The concept of an organ on a chip is a substantial improvement on the 3 R's of research: replace, reduce, and refine. Organ on a chip has not come out of the ether, but is the next stage in development of cheaper alternative research tools. It encompasses several different technologies or direct developments of these. The first is cell culturing. This is often a monoculture, a petri dish with only one cell type present, that is grown and the cells are allowed to divide. This is useful in some ways, but limited in others. The features of cell cultures are well known, as it is a common lab protocol. And next is lab on a chip. At a fundamental level, these are microcapillary tubes that minimize the reagents required for a specific function. The most common is cellular biology, but this process is two-dimensional, meaning that all of the cells are exposed to the experimental variables, and there is less complexity in interplay. The next stage was a 3D development of cell culturing. These are gel-based and more flexible as they can be cast or molded. This is still a stationary structure, but the design of the structure is significantly more complex. This means that fluid or non-rigid structures cannot be properly simulated, despite the fact that they can be custom-made. The fluid and dynamic nature of most structures in the human body or in an animal body cannot be recreated. Further, these are still mostly a monoculture or a monolayer. These two developments achieved 2 results important to organ on a chip. The first was limiting reagent use and creating a reliable process. The second is to demonstrate and prove the ability to culture cells on a solid structure and have these work in the assays that are used in organ on a chip. The benefits of organs on a chip are in 2 areas. The first is reducing the need for animal testing. By replacing the role of animals in testing, the moral and ethical dilemmas can be significantly reduced. It also reduces the problem of moving from an animal model to a human experiment, as the move between species can often carry a great many complications that mitigate the success seen in an animal model. The second is in reducing the cost of drug development. The ability to run more tests, more consistently, and with fewer staff reduces overheads. In the long term, this will reduce drug costs. Organ on a chip is the next stage in this, which goes from a rigid structure that is confined to a monolayer of homogeneous cells. The culture now encompasses several layers of different cells and makes mimicking structures like the vascular system, kidneys, and more possible. The multiple cell types and layers allow the cells to interact in complex ways. It makes physical barriers, modifications, and events viable. Examples include limiting the quantity of nutrients, introducing drugs and metabolic waste byproducts, and more based on the accessibility of the cells. This is a drastic increase to the complexity and possible interactions in a cell culture. There are a number of different organs on a chip either available or close to being available whilst they're in development. They are lung on a chip, heart on a chip, kidney on a chip, and artery on a chip. This covers 4 of the major systems that are widely relevant to some of the most far-reaching conditions at the moment. Organs on a chip, like those described here, are limited by a range of factors, and these are the primary source of criticism. Organs on a chip work on a similar basis to RAM cards. The chip is a polymer form that holds a large number of small channels that resemble the layout of organs. This is very much like the threads on a chip. By mimicking the layout and cell population, the chip can then emulate function. This collection of channels and cells is supported by a synthetic vascular system which supplies nutrients and other necessities. The structure of this chip allows the researcher to apply physical forces like blood pressure, mechanical motion, or even breathing. This is why organs on a chip work. In a more simplified explanation of why they work, the chip takes the cells and lays them as a number of layers. The easiest example of this is the lung on a chip. On both sides are alveolar cells with different purposes. These are separated by a third channel made of a membrane. These three channels allow different gas concentrations to move, but this also affects pressure. This then changes how the cells respond and what results are produced from this model of the gas exchange in the lungs. A more thorough explanation can be found in the description box below. The limitations of organs on a chip can be summarized by the design. It is a thin cross-section of an organ. This means that it only incorporates a limited part of the organ. Particularly problematic examples include the liver, where metabolites are produced and diffuse across a gradient, which means the whole organ may not function the same way as a limited section would. A second problem is the translucency of the chip. In the case of exposure to natural light, the UV radiation can cause denaturation or damage to the chip and experiment. The chip has a very small volume, and this limited capacity means the volume of a reagent or concentration is less important than the cells which cover the inside of the capillary. This also means the fluid content of the capillary do not mix. Finally, and perhaps most important, is that the chip limits the possible scope of the experiment. At present, an animal model can test a range of factors and be proved through several methods. Broadly speaking, these are proteomic, genomic, and changes to the environment. Examples of these methods include PCR, knockdowns, protein gels, and drugs. Examples of the utility of animal research include producing a range of organs and tissues, which each need separate chips if you were to use a chip-on-method approach. These chips may not reflect the complexity of the whole organism. For example, blood drawn from a mouse may contain a number of things that you cannot get from a chip, including neurotransmitters that do not originate from the vascular system. Considering the growing range of organs that can be simulated, the next stage of organs on a chip are myriad, but the most likely candidate is something called human on a chip. This addresses some of the problems found in single or limited organ systems. By combining multiple organs into a multi-planed and consequently multivariate experiment, the increasingly complex interactions can be modeled to some degree. By comparison to the early development of organs on a chip, these humans on a chip are still very much in the stages of infancy. Whilst the development of organ on a chip is a great step forwards in the 3 R's of research, it is not a perfect replacement in the case of animal research. It fails to accommodate the whole organism problem, the interaction of multiple systems, and the complexity of an internal variation within one organ or system. Thank you for watching this video. If it has been of interest, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.